Hi and Assalamualaikum.
Welcome back to our
e-channel.
Today's video is about the microarray technology in breast cancer studies.
Before we proceed, what is microarray techniques? Microarray is an orderly arrangement of samples where matching of known and unknown DNA sequence is done based on base pairing rules. This array uses common assay systems such as microplates or standard protein membranes. The sample spotted size are typically less than 200 micron. The sample known as probe are immobilized on solid support, for example, on a microscope glass slides. The spot can be DNA, cDNA, or oligonucleotides. This technique allows parallel analysis for gene expression and discovery, as it used to determine the complementary binding of unknown sequence. There are three types of microarray. First, microarray for expression analysis. Second, microarray for mutation analysis. And the third one, comparative genomic hybridization. For this video, we are focusing on DNA microarray, which is the microarray for expression analysis.
DNA microarrays are used to see which genes are activated or which genes are repressed. Every gene are compared simultaneously. In the video, let's compare the pattern of gene expression in breast cancer tissue compared to the normal tissue. When a gene is expressed, it is transcribed into mRNA. The mRNA from the two tissues are isolated and converted into complementary strand of DNA called cDNA. This process is by using the enzyme that is known as reverse transcriptase. To distinguish the pools of the cDNA, the molecules are fluorescently labeled red for the tumor-derived pool and green for the cDNAs derived from the normal tissue. DNA microarrays are used to compare the cDNA samples. DNA microarrays can contain 60,000 or more different DNA sequences attached in microscopic spot to a glass slide. The different DNA sequences are oligonucleotides of about 20 bases in length. The cDNA samples are mixed together and added to the microarray. cDNA that are complementary to the oligonucleotide on the microarray will bind or hybridize with the DNA. Unbound cDNA are washed away. A scanner detects the patterns of hybridization by sensing the fluorescent signals. In this example, the red spot indicates the expressions of the gene is higher in the tumor tissue compared to the normal tissue. In contrast, the green spot indicates the expressions of the gene is higher in normal tissue than in tumor tissue. The yellow spot indicates that the gene is expressed equally in both tissue. If the gene is not expressed in either tissue, the spot will not fluorescent. This is because each area of the microarray contains a known sequence that is corresponding to a known gene.
Next, let's have a look on protein microarray, an example of microarray that is used in breast cancer study. Protein microarray is the simultaneous analysis of a large number of different proteins in one single experiment, allowing for a parallel assessment of a large number of proteins. Protein microarray is used as the general term for the high-throughput parallel chip investigation of protein identity, quantity, interaction, and function. There are two general types of protein array, the forward-phase protein array and reverse-phase protein array, but in this video we are focusing on forward-phase protein microarray. For forward-phase protein microarray, the elements on the array are captured molecules. As previously mentioned, this can include antibodies, proteins, nucleotide, or aptamers. Of the different types, the most common one is the antibody array. Antibody array, as the name suggests, take advantage of the specific interaction between antibodies and proteins and use antibody as the capture or bait probes. With the array of capture probe, the analyte such as cellulose lysate or serum sample is applied and incubated on this surface to allow interaction between capture probes and the analyte. There are two general categories of detection formats, label-based and sandwich. Label-based method require pre-treatment of the sample prior to incubation in the microarray. The analyte can be either directly labeled with fluorophores or quantum dots, or indirectly tagged after capture with the secondary labeled antibody. There are a few papers on forward-phase protein array. One of them is from Lin, which is use a forward-phase array of antibodies for cytokine to detect the protein expression levels of different cytokines in breast cancer. This helps in identifying the important role of interleukin-8 in breast cancer oncogenesis.